Good afternoon, Beacons. Welcome to the virtual faculty research celebration. My name is Marcelo Suarez Orozco, and I am deeply, deeply honored to serve as the new chancellor of the University of Massachusetts, Boston. I am pleased to be here today as we highlight and celebrate the accomplishments of our esteemed faculty in their contributions to the areas of research, scholarship, and innovation. And above all, to thank them for their support and mentorship of our students. Since 2006, research at UMass Boston has grown significantly from over $25 million in research dollars in 2006 to a high of over 70 million in 2017 and 66 million just last year. Areas of research strength, from community engaged projects to the very, very frontiers of basic research, include developmental psychology and brain sciences, green and environmental chemistry, climate change impact on coastal communities, aquaculture, biomedical sciences with a defined focus on cancer, gerontology, population and exercise health, sustainability, quantum information and quantum computing, remote sensing, early childhood and STEM education, data sciences and artificial intelligence, cyber security, nanoengineering, to name just a few. These are among the defining issues of our time. The theme of this year's event is the teaching soul, research and mentoring. The teaching soul, research and mentoring. Today's program will showcase two of our faculty members who are working with two inspiring, amazing students who are supported by the Beacon Student Success Fellowship Program. Recognizing the value of experiential learning, the Beacon Student Success Fellowship Program was launched in the spring of 2014. The fellowship provides UMass Boston students with financial support for experiential learning activities such as internships in nonprofit or government organizations, volunteer work, service learning, civic engagement projects, mentor research experiences, participation in academic symposia, and travel, national and international, related to academic projects of study that take place during the summertime. Today, we not only celebrate our faculty's commitment to research across their respective disciplines, we also celebrate the mentees who have benefited greatly from the relationships with their faculty and whose lives have no doubt been changed for the better for the individual and unique guidance they have received in preparation for their careers in the sciences, in public policy, among many, many domains. Employers today value the experiences as much as they do solid academic foundation. Experiences that at the University of Massachusetts, Boston, as our students we're celebrating today, and our students presenters will show, this is an opportunity at UMass Boston to obtain both a solid academic foundation and hands-on experiences in their various domain of scholarly study. This afternoon, we will hear more about the uplifting, the fundamental nature of the mentor-mentee relationship. We will listen to two faculty members talk about how their students inspire them. And we will hear from our students about the ways they have been able to begin mapping out their futures thanks to the support of their individual faculty mentors. In a moment, Provost Dr. Emily McDermott will introduce our presenters. Dr. Hannah Sevian, Professor of Chemistry, and her mentee, Daniliz Capellán Pichardo, who is studying chemistry and will graduate this December. Congratulations to Daniliz. They will give a presentation entitled Connecting Chemistry to Life Research. We will also hear from Professor Kellee Siegfried and her mentee, Keith Ameyaw, who is a biology major and will graduate in May 2021. Congratulations, Keith. They will give a presentation entitled Male or Female, How DMRT genes regulate sexual fate and fertility. As we prepare to get underway, let me remind you that this is a celebration of research and relationships at their best. How much more powerful both are when joined through the prism of mentorship in an academic setting. I know personally how my mentors, when I was in college, shaped my intellectual curiosity and indeed my life trajectory. These relationships from both the faculty and the student perspectives ensure that members of the University of Massachusetts Boston community make their mark in the Commonwealth, the nation, and the world in the most meaningful ways possible. Thank you again for joining us and for all you do on behalf of our university. Congratulations, Beacons. And now it is my pleasure to introduce our interim provost and vice chancellor for academic affairs, Dr. Emily McDermott.
Welcome to the Fall 2020 Faculty Research Celebration with its traditional focus on the teaching soul, research and mentoring. At universities, we sometimes speak of teaching and research as separate aspects of a university's mission. But when we do that, we are in danger of inadvertently blurring the reality that research and teaching lie on a continuum. We create knowledge and we share it. We share knowledge to allow others in turn to create it. Over the years, this campus has worked diligently to connect the ends of this continuum in recognition that an important aim of a university education is to light the flame of lifelong learning in our students. Introducing students to research as early in their studies as they are ready for it is an effective means of achieving this aim. In its simplest form, research promotes a recognition both that not all questions that need to be posed have yet been posed, and that not all answers that are needed are contained in textbooks or Wikipedia. Research is a catalyst for curiosity, just as, conversely, curiosity is a catalyst for research. Today you will hear from two of our undergraduate researchers who have learned these lessons while working under the guidance of their UMass Boston faculty mentors and in collaboration with full research teams. The first talk you will hear, Connecting Chemistry to Life, involves the work of Daniliz, Capellán, Pichardo, a chemistry major, mentored by Professor Sevian. An important focus of the research efforts of Professor Sevian's lab is study and promotion of ways to enhance access and opportunity for all to learn chemistry. Working with learners at all levels, Professor Sevian and others on her team highlight the unique value of the discipline of chemistry, featuring inquiry-based activities to promote learning and to train a new generation of researcher teachers. Daniliz structured her undergraduate chemistry thesis around the question of how students in organic chemistry use socially derived constructs of a molecule when solving practical problems. This will be the topic of her talk today. The second talk you will hear, Male or Female, How DMRT Genes Regulate Sexual Fate and Fertility, is based on the work of Keith Ameyaw, a biology major, and his mentor, Professor Kellee Siegfried. Dr. Siegfried's laboratory investigates genetic regulation underlying development of the gonad, investigating specifically how differences between the sexes are manifested during development, processes necessary for fertility and reproduction. Working with the Zebrafish model system, Keith became involved in this work through the Initiative for Maximizing Student Development program. IMSD is an NIH-funded year-round research-intensive, skill-building mentoring program for undergraduate students interested in pursuing research careers in the biomedical sciences. Keith himself is planning a career as a physician scientist. I'm sure you will enjoy today's celebration.
Good afternoon. Thank you for welcoming us to this celebration. We're very excited to be here. Daniliz and I are going to be presenting about connecting chemistry to life, and we are grateful to Provost Emily McDermott and to Chancellor Marcelo Suarez Orozco for introducing us and for hosting this event. The photographs that you see on the screen are going to be explained a little bit as we move forward.
Hello, my name is Daniliz Capellán Pichardo. I was born in the Dominican Republic and I immigrated here when I was 3 years old to New York City, and I moved to Boston for college. I'm a chemistry major and I joined the Sevian research group in 2018. I'm also a McNair Scholar and I am a BSSF recipient. I've also worked under NSF grant from Dr. Sevian's lab, and I was also part of the UTeach program as a freshman. Here is a picture of one of the last times I went to the Dominican Republic of my family. And there's a picture of a frog in the middle because it's not uncommon to find frogs anywhere that's cold and dark. So you would find them in the bathrooms and the like. And so I've always been interested in biology. And so instead of, and science, instead of just killing them, I would grab them and walk them down to the river that was only a little means away from our house. Even if I found 100 in one day, I would walk them each time because I couldn't bear killing them.
And I'll tell you a little bit About myself, I was born here from an immigrant family, and there were multiple languages spoken in my home. I studied chemical engineering. Like many of our students, I lived at home and commuted to campus and also worked full time. My initial background was in theoretical chemistry, but then I moved to teaching high school, and I taught in the Spanish bilingual program at Chelsea High School for 7 years before I joined UMass Boston in 2001. The focus in our lab is on studying a disciplinary nature of science, specifically chemistry, and learning as culturally relevant social activity. So you will hear a little bit about Daniliz's work, in particular working on socially relevant social activity or culturally relevant social activity and studying how students are thinking about chemistry. The photograph that you see on the screen is taken in Yerevan, Armenia. It's the eternal flame from the genocide memorial, where I was fortunate to visit with my family 2 years ago. This is another of my favorite photographs. This is the classroom where I normally teach. This is 1100 University Hall, and this is where I met Daniliz in 2016 when she was taking General Chemistry 1 with me. At that time, there were 230 students or something in the classroom, and it's the off-semester sequence of general chemistry, which I've had the great privilege to teach for almost 20 years at UMass Boston. One thing that I love about chemistry is that it's one of the oldest disciplines, and it grew from the practical arts. Its purpose has always been to improve the human condition, and that makes it very easy to relate chemistry to life. So in other words, who people are really matters in chemistry. Here's a typical example of something that, that we talk about in the chemistry classes. I'll be teaching this in about 2 weeks in the General Chemistry 2 course that I'm teaching remotely now. So you see here two versions of the molecule of cocaine. One is the base version and one is the acid version. They can both be taken by people, and they have different bioavailability. So if you'll please take a look on your screen, there will be a poll appearing shortly. I'll expect you to do a calculation. And I'm just kidding. Seriously, the whole point is that students can see themselves in chemistry, and they can relate this to their lived worlds. Many of the students who take general chemistry are planning on going on in the biomedical sciences, and this is something that's very relevant for them, for who they may end up working with. So I was really, really pleased when Daniliz asked me in early 2018 if she could join my research group.
Yeah, so I'll say that in traditional lecture halls, we learn about research that was done, hundreds of years ago. But in Dr. Sevian's class, we learned also how it applies to my life. So here, there's also a project timeline of the work that I've done since I joined the lab. So I learned how to operate an eye tracker, which is amazing. I was able to take a graduate chemistry course on activity theory, which actually helped me what I learned there, I used as a framework for my current research. I was able to learn how to develop a methods and apply it to my project. And I also learned some basic R coding, so I was able to use it for data analysis that I'm still using to analyze some of the data. And so with all of that, I was able to write my capstone thesis, which I presented to the chemistry faculty last semester. And my paper was also accepted to ACS, although I wasn't able to do it because of COVID, but it was a great experience.
So you see here a schematic of an eye tracker. The orange or the yellow prism is actually lights from 2 infrared cameras that are looking into the person's eyes and able to triangulate and figure out what the person is looking at. And we use eye tracking to look at both 2-dimensional and 3-dimensional representations of molecules so that we can learn how students are thinking by what they say as well as what they're looking at. What people pay attention to, it helps us to understand how people use their chemical thinking to solve problems that are unique to chemistry, that chemistry allows us to answer. We use a theory called the conceptual profile theory, which builds upon the heterogeneity of thought from a sociocultural perspective. And Daniliz is going to be talking about that a little bit in an upcoming slide. But first, she's going to talk a little bit about the relevance of this.
Yeah, so this helped me a lot because it helped me to really be more conscious of how I think about chemistry in different contexts. And also because before this, I was really thinking about being a chemistry teacher. And so having this experience, I was able to, I realized how much I really love doing research, asking questions and solving them, looking for the answers. And so I'm planning to continue to extend my knowledge of research, and going to graduate school to study more. And I also realized that, chemistry can have other purposes, especially chemistry education from a social justice perspective. And so that's something that's very interesting to me as well. So to explain a bit about how conceptual profile works, conceptual profile theory is pretty much it describes how there's heterogeneity of thought in different ways of thinking. So each person has their own way of seeing and conceptualizing the world to signify their experiences, depending on their different cultural backgrounds or the different contexts that they may be in. So the zones that we talk about, or different modes of thinking in a conceptual profile, are the meanings for a concept that are stabilized by three commitments. And so this allows us to have a more holistic approach when we're looking at student responses. Instead of separating student discourse into right or wrong, we accept that there are various ways of thinking and speaking depending on different conditions. So if we, to give an example, if I were to ask you all, what is the meaning of life? We don't all have a different response depending on your context, perhaps your religious background, your cultural background. If I were to ask this in a biology class versus a philosophy class. So we are here we are aware that these differences exist and that they are contextually dependent, and that helps us to focus more on the relationships between visual representations and student thinking. So the two main questions we want to answer for this research project were, first, do organic chemistry students attend or look at different molecular features when questions trigger them to rely on different zones? And two, do the student zones shift when they're answering chemistry questions? So on the bottom, we see an example of one of the questions that we asked. So if you were to mix this with water, would it dissolve? And how do you explain that? So the molecule in question is Viagra, and the points that are highlighted in red are areas that we focused on to see if students would look at these specific points. So what we found were that 3 of the 4 zones described in Dr. Mortimer's book were used, which was to be expected because we were only trying to trigger those 3 points. We also found that as the complexity of the molecules increase, the reliance of the zones changes. We also found that students tend to look at the molecules differently depending on the zones that are being used. And so in the future, we hope to analyze more on what the sequences of these areas of interest are and if they are consistent between students or between zones being used.
So at this point, we would like to really acknowledge the people and the funding that have helped to support this work that Daniliz has done. In particular, we would like to thank Jessica Karch, who you see here on the bottom right. She is a 5th-year PhD student in the lab. She is also an NSF Graduate Research Fellow, and she's been the primary mentor to Daniliz. Also in the back here, you see Ameyaw, who is a visiting scientist from Pernambuco, from Recife in Brazil. And she was here for a semester and she taught the course on activity theory, the graduate course, which several of us in this photograph took, and also Daniliz took this graduate course in chemistry. We're very grateful to both the National Science Foundation, the Beacon Student Success Fellowship, McNair, and all of you. Thank you for joining us today. At this point, we ask that you, if you have questions that you would like to bring up, to please place those in the Q&A section. And we also asked originally that the chat remain open because we also feel very much isolated all of us in Zoom. So we thought that it might be nice for those of you who know each other to be able to chat with each other, but please keep them separated. If you have questions, put them in the Q&A and indicate whether they're for Kellee and Keith or for Daniliz and me. And now we turn to Kellee and Keith, who will present next.
Okay, hi. Thanks for this opportunity to participate in this event here. Keith and I are both very excited to be involved, and thanks to all of you for being here. So I am going to start off and introduce the project that Keith and I have been collaborating on, and then Keith will come in and talk about his specific part of this project. So I first want to just give an overview of the project that he's involved in. Okay, so in my lab, we're interested in processes related to fertility, and specifically, as you heard in the introductions, we're interested in how you get differences between the sexes during development, and also how do we produce the eggs and sperm and continue to produce those to maintain fertility throughout our lives. And Keith and I are going to talk a little bit about this first question, in terms of how the differences between the sexes arise. So we're looking at this with respect to the reproductive organs, the ovary and the testis. And as you can see in these drawings of the human ovary and testis, these organs develop with very different structures and functions. For example, the testis is going to be specialized to produce the sperm, whereas the ovary is specialized to produce the eggs. And in addition, they have very distinct endocrine functions as well. And so understanding the development of these organs and how you get a manifestation of either ovary or testes is a really important aspect of investigating infertility and understanding infertility, as well as endocrine function. And fertility is a major problem today. So around 15% of couples who wish to have children experience infertility. And largely these causes are still unknown, although many of them are suspected to be genetic, which is something we're very interested in looking at. So our main question that we're gonna be looking into today is what are the genes that control the sex-specific development of the ovary and testes? And so first, I want to talk to you about the organism that we use. We're using a model animal to study this question called the zebrafish. So the zebrafish is a great model to study, experimental model to study development in both humans as well as other fish species. And when I mean fish in fish, I mean we can apply what we learn in zebrafish to more commercially relevant fish, such as those that we eat. With respect to human disease, the zebrafish shares about 70% of their genes with humans, and these genes typically have the same or similar functions in both fish and humans. And so we can use the fish to study genetic causes of diseases in humans, which is the approach we are looking at also with respect to fertility. So our investigations of zebrafish in this process started out with looking at a gene called DMRT1, which we found to be important for testes fate determination and development. And this project was largely led by a previous PhD student in the lab named Caitlin Webster, who graduated a couple of years ago. And what we found in this project is that the DMRT1 gene is really important for testis development. So as you can see here in this representation is in normal wild type fish, you have males and females in the population at approximate equal ratios typically. However, when we create a mutation that disrupts the DMRT1 gene so that it's no longer functional, what we found was that largely the fish were developing as females and we only rarely saw males. And just to show you how the data looked, actually, This is the two columns on the left are showing the normal or the wild type ratio of the two sexes, the males and the females. And as you can see in the DMRT1 mutants, we had a huge increase in the ratio of females relative to males, and only around 10% of males were present in this population. So our further analysis of these mutants, we saw that most of the DMRT1 mutants became females, and those females were fertile, which told us that DMRT1 is not important for the ovary. We did get a few DMRT1 mutant males, as I said. However, these males were all sterile. And we saw that DMRT1 was necessary for the development of the testis and the production of sperm. And so what we became interested with respect to these data is how we get these small number of males that lack a DMRT1, a functional DMRT1 gene. So how can the fish still become a male when it does not have this important regulator of male sex and male testis development? And so we came up with a hypothesis where we thought maybe the other genes that are similar to DMRT1 might also have a role in this process and can carry out the male determination in the absence of DMRT1. So we're looking at whether or not genes that are similar to DMRT1 also have a role in testis determination and fertility. And so the DMRT1 gene is part of this family of genes. Here's DMRT1 here. There's 5 of these genes in zebrafish, and Keith is going to talk to you about his work on the DMRT2B gene. So this was a big undertaking. We decided to investigate this entire gene family and their role in regulating ovary and testis development and fertility. So this project is led by a current graduate student, Jocelyn Steinfeld, and she has worked with a large number of graduate students over the last few years. Here's Keith here, who's going to talk to you next. Currently, Keith and Chris are in the lab. Jessica has moved into the PhD program and is still in the lab as well. And so this is a large project that really wouldn't be possible without all of the involvement of these undergraduate students who have contributed to this body of work. And I just want to point out that the students have also been really successful in attaining various fellowships to help support themselves and their research, including the Beacon Student Success Fellowship. Keith has also himself, in addition to that fellowship, has gotten a fellowship from the College of Science and Mathematics Research Fellowship, as well as is supported by the IMSD program. Okay, so with that, I'm going to pass this on to Keith.
Hey guys, well, to start, my name is Keith. I am a senior at UMass Boston studying biology and minoring in sociology. So a bit about how I got into research. So after the spring semester of my freshman year, I applied to the SPARK program, which is a program with a partnership between Dana-Farber, Harvard Cancer Center, and UMass Boston. So I had the privilege of joining Dr. Alon Klein's lab, where we studied how cell fate is determined. So using the zebrafish model, we studied how we can couple cell cycle and cell differentiation and division. So one thing that I really stuck with me during that lab is kind of like a fun fact that one of the graduate students gave me. And it was like, oh, do you know that zebrafish, sex determination is determined by, numerous of factors, which I found to be very interesting and quite curious about it. So I did some digging and I stumbled upon Dr. Siegfried's lab, where they also, studied zebrafish sex determination and differentiation. So I was curious about that, emailed her, and she welcomed me. So to the fall of 2018, I joined her lab, and under the mentorship of Jocelyn Steinfeld, we studied characterizing the role of these DMRT genes that Professor Siegfried talked about in zebrafish sex determination and fertility. So one of the previous work that was done by my mentor, Jocelyn, was that, okay, so if we're trying to understand if these DMRT genes were expressed or were responsible for sex determination and differentiation, why not check where they're expressed? Are they either expressed in the testes or the ovaries? Because that kind of gives us a general idea of what these genes could be doing. So we tested these 5 genes, DMRT1, 2A, 2B, 3, and 5. And it turns out that they're actually expressed in both gonadal tissue, the testes and the ovaries. So my work specifically, in addition to that, was to move further and test to see if these genes were also expressed in different tissue panels, such as the kidney, brain, eyes, heart, skin, liver, and the viscera. So actually it turns out that these genes have a varying tissue expression profile in different tissues, with the exception of DMRT1 and DMRT3, which are gonad specific, meaning that they're only expressed in the testes and ovaries. So that kind of gives us, or even other labs, like an idea of like where these genes could be playing a role in the zebrafish body. So to focus in on one of these genes, the gene that I focused on was DMRT2B, and how we asked this question on if it played a role in zebrafish gonads. So how to test for that was we generated a DMRT2 mutant, in which the first experiment we did was to kind of see if a mutant or a loss of this gene causes a difference in the sex ratio of a population. So this figure on our left shows us our wild types, which which are our normal phenotypical one that still contains the DMRT gene, and you can see that it's mostly male biased. But when you come to the mutant, that's also male biased. So kind of telling us that DMRT1 or DMRT2B, sorry, does not really change the sex ratio of this particular family. In addition to that, we did a fertility assay in which we're checking to see if these mutants that were produced with this line were able to give rise to other zebrafish, or able to kind of fertilize and give rise to other zebrafish, which we checked for the female mutant and also for the male mutant, and we checked that those were also normal compared to the wild type. So we concluded that DMRT2B does not play a role in sex determination or fertility, like how DMRT1 did play a role. So my next experiment or project was really to see if DMRT2B was redundant with the functionality of DMRT1. So if you recall, Professor Siegfried talked about how DMRT1, a loss of DMRT1 in a family kind of produced a female biased population, meaning that DMRT1 was important for male fate. So with this figure, we can kind of divide it into 2 subsections, A and B. And in A, these are our non-DMRT1 mutants, meaning that they wouldn't show the same phenotype that DMRT1 mutants have. And in B, we have our DMRT1 mutant line, in addition to the double mutant with 2B. So treating our A as our wild type, you can kind of see that this family in general is male bias. But once you introduce the DMRT1 mutant in portion B, you can kind of see there's a female biased tendency going on, which, as you can see from previous slides, you could see, you already know that DMRT1 is female biased. So it was, kind of confirming what we already knew. But when we come to the last section of figure B, where we have our double mutant, we kind of assume that something, or DMRT2 would have a role or would show redundancy with the functionality of DMRT1, but it actually turns out that DMRT1 doesn't have any role or any redundancy towards the functionality of DMRT1. So with that too, we also concluded that DMRT2B was not required for sex determination, nor was it redundant with DMRT1. Okay, so to summarize these 2 slides of data, the first one talked about how DMRT1 didn't play a role in sex determination or fertility in zebrafish on its own. And also we knew that with DMRT1 was also not redundant with the functionality of DMRT1. So why is this research important? You may ask yourself. So as Professor Siegfried also talked about 15% of couples suffer from infertility, and also 40% of these causes are not known. So this research goes a long way in understanding what mechanisms kind of govern fertility and determination and sex determination. So a bit about where this research has taken me. So over to your right is me in ISC, and this was, I think, my first exposure to present in my research. So it was a great segue, which led into that fall with me attending the annual biomedical research conference for minority students in 2019. And through IMSD, I was privileged for them to pay for my expenses to have that opportunity to go and present. And with that conference, I won an award, which was very, very kind of a confidence booster in a way. It gave me kind of this notion that I could pursue a career in research, which was glad. And one thing that I really appreciate from research is kind of like the family you built around it. These mentors are really kind of like a guideline for you to move on and also move past. I often equate it to life in a sense. It allows you to use what you learned in class to kind of like, move on, to like kind of think critically about things in life. Okay, so that is it for me. Big thank you to the Siegfried Lab and Jocelyn Steinfeld, who was my mentor, and also to the IMSD program. Thank you. I believe we have a question.
We have one question. I'll read the question.
Okay.
So the question is the DMRT1 gene also found in humans? And if so, have they found any evidence of it affecting human infertility? Keith, do you want to answer it?
Yes. So
if you like, I can answer it.
Okay. Yeah. So yeah, most of the DMRT genes, there's actually, I believe, correct me if I'm wrong, there's 8 DMRT genes in humans, and DMRT1 is conserved among vertebrates, which includes humans. Is there any evidence of it affecting human infertility? I'll pass that to Kellee. I'm not sure.
Okay. Yeah, so DMRT1 is, there are mutations that disrupt it in humans that lead to what are called XY sex reversal disorders of sexual development, meaning that these are people who should develop with a male phenotype, a male physical attributes, and develop a testes, but instead have some female characteristics, and that does result in many different problems, including sterility, as well as increased chances of getting cancers in the gonad as well, in the gonad tissue. And there's also some endocrine problems related to that, because the gonad is an important endocrine organ, and its development is disrupted when that gene is affected in humans. So yeah, it is very important in humans as well. Keith, you have another question from Andrew Grosofsky. Are DMRT genes involved in other pathways of cell fate determination other than the gonads?
That's an interesting one. We could talk about the paper that we did for lab with the double mutation with CYP19. So I don't know if that's like an answer to you, but there was a paper that was discussing how the pathway between, or the mechanism between, how CYP19 generates estrogen in the ovaries, how that could be affected by DMRT1, or how CYP19 actually kind of inhibits DMRT1 in order to generate female development.
So I think Andrew's also asking about other DMRT genes, not only DMRT1.
Oh, okay. Yeah, so that's what we're trying to study. So this one was on 2B, DMRT 2B, but we're also working on DMRT 2A, 2B, and 5, and we haven't yet kind of fully done all those projects. But we're also working on again those double mutants between DMRT1, which we know drives male fate, and also other DMRT genes.
Yeah, and I'll just add a little bit to what Keith has said, is that outside of the gonad, there are some roles of other DMRT genes as well in development. For example, in regulating your left-right asymmetry is something that has some of the DMRT genes have been shown to be involved, which is important for heart development, because the way your heart develops requires that you have a left and a right side that's specified. So that's just one example. DMRT3 has been shown to be involved in some development of the skeletal structures like the ribs. So as Keith showed, you know, some of these DMRT genes are expressed in other places in the body, and work in mainly in mouse, and some mutant data from humans shows that they do have some roles outside of the gonad.
And also DMRT5 is expressed in the brain. I think it's lethal if it's not knocked out.
So I'm unable to type in the question and answer, so I'm just going to ask my question because Keith brought it up. You said that DMRT is expressed in the brain, and I know that there are all sorts of psychology functions, and you said that you're a sociology major, so I think there's some relationship, right? So are there things that DMRT expression does to how the brain develops in different parts that are in the left and the right?
So I believe for the left and right asymmetry, I think that's DMRT 2A. I'm not sure how the mechanism for that works particular. And for DMRT 5, I know that it's lethal because I think it's responsible, or it plays a part in brain development. But I don't know how the mechanism in that works as to how far it goes in that.
I can add another interesting factoid about DMRT3 roles in the brain in horses, which is a funny kind of thing that has come out in a few years ago, where there's some horses that walk with certain types of gait, either, you know, I don't even know if I can do it, but they either kind of do like this, or the 2 feet kind of go like this. And that actually it can affect how fast they run. And that's actually regulated by DMRT3. And so there's some body of research interested in DMRT3 and these different ways that horses walk or run, which is also something that's happening in the brain. We have another question from Jens Rister, who's asking, is there a homolog in flies that affects the horse's gait? So the DMRT3, he's asking, there's not a direct ortholog of DMRT3 in Drosophila. So no, there's I don't think that will have a similar role in Drosophila.
If nobody has more questions, I guess I would just say what a wonderful presentation, or wonderful 2 presentations these have been, and how exciting it is to see our faculty providing such wonderful mentorship to our students, and our students doing such a good job in return of building their skills in the lab, and their curiosity, and their ability to do wonderful research, and go out and make us all very, very proud as you leave the university.